I got so much motherfucking design on right now.
Break it down, bring it back, make it clap. I got 3 old thoughts, new block, where that day. Pictures on my line, and you know
Hi and Assalamualaikum everyone. So this video will be about our field trip session in the forest behind our faculty. So before we start, I will introduce my group mate first. The first group member is Nurizati binti Rabbani. The second one is me, Nurin Ain Sofia. The third one is Shahida Aida. And the fourth one is Siti Nur Nadira. Different species often inhabit the same spaces and share the same resources. Hence, they interact in variety of ways, which is called symbiosis. Symbiotic relationships are the close association formed between the two species. There are 4 main symbiotic relationships, which are mutualism, parasitism, commensalism, and competition. Okay, so what is the purpose of this field trip? This field trip is to find the interaction between the living organism in the forest, such as the connection and the interaction between the plants and the plants, or animals and plants. So in this field trip, we're going to find as much as interaction as we can.
For the methodology of this experiment, we were divided to several groups according to lab group to begin our assignment. The goal is to look around and select among many creatures present and apply what we have learned about community ecology.
In community ecology, there are various types of interaction among species. There is competition, predation, mutualism, commensalism, and parasitism. Firstly, competition shows that species may compete for resources such as food, water, or space. Competition can be intraspecific between the same species or interspecific between the different species. Furthermore, in predation, the predator consumes the prey. This interaction can influence population dynamic and can lead to adaptation in both predator and prey species. Meanwhile, mutualism interaction benefits both species involved. Also, in commensalism, one species benefits while the other is unaffected, and lastly, in parasitism interaction, involve the parasite benefiting at the expense of the host. Parasite can derive nutrition and shelter from the host and causing harm in the process. Moreover, relationship between Coccinellidae or ladybug and leaf is mutualism. Ladybug are the natural predator of plant pests including aphids. Plant pests can damage plant, so by having a ladybug patrolling plant can give benefit to the plant itself. In return, the plant indirectly provides the ladybug with a supply of food.
So the next interaction is between the ant, anthill, and the plant around it. So the interaction here is mutualism. Why? Mutualism is when the two organisms get benefits from each other. First, the plant get protection from the ant. Because why? Because the ant eat the insects on the plant leaf. So the plant will get avoided from the insects that will damage their plants, and the ants get food from the plant. So both of them give the benefits to each other. So it's a mutualism interaction. So what we have here, the first interaction is between this big tree and another tree on his tree trunk. So the interaction is parasitism. The another tree on the trunk is trying to absorb the nutrient from the big tree. So when another one gets benefits and another one have a negative impact, it is called parasitism.
The next interaction between organisms that we found was the interaction between Asplenium, Nidus, and also tree. As we observe and we do research about them, the type of interaction that happened between these two organisms is commensalism. Asplenium nidus depend on tree for shelter in order to survive, but tree get nothing from that organism. So this is what we call as commensalism, which is one side of the species gain benefits but the other side neither gain cause nor harm. As we research in deeper about these two organisms, we found a fun fact about Asplenium nidus, which is it spread by spores rather than seeds. The next interactions are between Isoptera and Formicidae. These two organisms have two types of interactions, which is the first one is competition and the second one is prey-predator. The first one is competition. This may happen when termites and also ants are compete for the same shelter or the same nest. So this may affect the diversity and distribution of whether ants or termites. This is because if one of them lose from the competition, so they need to migrate to other places. So when they migrate, distribution happen. The second interactions between termites and also ants is prey-predator. Ants usually will hunt for food by attacking the termites colonies and eat them.
Or commensalism relationship between insect and plants. Nodules on leaf or leaf guards are generally caused by insect or other foreign organisms such as bacteria, fungi, mites, nematodes, and even virus. Next is mycorrhizae relationship. Mycorrhizae are symbiotic relationships that form between fungi and plants. The fungi colonize the root system of the host plant, providing increased water and nutrient absorption capabilities, while the plants provide the fungus with carbohydrate formed from photosynthesis. Next, mutualism relationship. It is between Formicidae or ants with plants. The plants will provide food and nesting space for ants that provide them against herbivores.
In addition, Perosia piloceloid or dragon scale are found growing on trees. The type of interaction between these two is commensalism. Perosia piloceloid having a rooting site without harming the host tree.
Alright, you already know about the interactions that we found during our field trip. So now I'm going to conclude with the benefits that we obtained from our field trip. The first benefit is that this field trip exposed that there is richness of biodiversity in forests at Kuala Pilah. Before the field trip, we never realized how much species that could exist in Kuala Pilah, and after field trip, we realized that there's a lot of species that we need to discover more, and it's very interesting. The second benefit is that students able to differentiate between the different type of interactions in real life. The third benefit is that this field trip teach students to be more cautious to be in forest in order to maintain the sustainability of the forest. And the fourth benefit is that it makes students able to explore the nature, which is not all students experience it before.
In this experiment, we learned about normal distribution and its parameter, which is the mean and variance, and initial understanding about correlation. The objective is to calculate and describe the following descriptive statistics, which is mean, variance, and standard deviation. Second, to understand the concept of correlation. And lastly, to determine the relationship between the two variables. For the methodology, firstly, 10 leaves were taken. Their length and width was measured and recorded. The mean, variance, and standard deviation of the length and the width of the 10 leaves was calculated. Second, another 10 leaves were taken and the first procedure was repeated. 3 sets of leaves were observed. The result was tabulated. And lastly, the data of 30 leaves was plotted in a graph of leaf length versus leaf width. The relationship between length and width of the leaves was identified and discussed.
Then, I'm going to continue on the result parts. In this part, I'm going to discuss about the 4 main components, which is the first one, the set of data, and then the second one about the calculation of mean, variance, and standard deviation. The third one, interpretation of graph, and the last one, I'm going to talk about the regression and also correlation. But before that, we need to know about the formula that being used in this experiment. So basically, the symbol in this formula Let me explain. Okay, X represent mean, SI represent the reading of measurement leaf, and then N represent the number of leaf being used in this experiment. We collected 3 set of data for this experiment, which is involved 30 different leaf, 10 leaf per set. This is result for the first set. This is result for the second set. And this is result for the third set. For graph interpretation, we plotted this graph by length versus width with all 30 readings that we obtained from different leaf. Based on plotted graph, this shows positive correlation with an increasing line like this. Then, this is the regression and correlation table that our group made from Excel. From this, we obtained a regression value of 0.68 and also the p-value of 0.000875. This shows that the p-value is lower than 0.005 and indicating that our results are significant.
So for discussion part, the value P obtained is less than 0.005. This means that the value obtained is significant. When the value obtained is less than 0.005, it is concluded that the difference exists but there is no actual difference between it. There are differences in the mean of lengths between the different set of leaf. The value of mean, the length of the leaf for set 1 is 13.32, for set 2 is 11.42 and for the set 3 is 11.47. The mean value of each set is different because the leaf will pick randomly from different tree of the same species. Hence, the size of the leaf is not constant and different in size. The length and the size is different might be because of the tree response to different light environment. For example, leaf on the upper part of the plant receive more sunlight compared to the lower part of the tree. So, the leaf of the upper part of the tree is bigger. There are also differences in the mean of width between the three sets of the leaf. The mean value of the width of leaf for set 1, set 2 and set 3 is 5.88, 4.85 and 4.64 respectively. There are only a slight difference between the mean value. This is because the width of the leaf depends on the nutrient absorbed by the tree. The more the nutrient absorbed by the tree, the more the width, the more the growth and the development of the plant, so the leaf will be bigger.
The second question is, what is meant by variance? The leaf have different measurement of width and length. What is the meaning of this statement? Discuss with respect to variance and what is meant by standard deviation. In view of distribution of all data points in a set, variance is an indication of how individual data points differ from mean values. This is the mean squared difference between each data point and its center of distribution. In the experiment, 10 lists of each 3 sets with 30 leaves in total is resulted in different measurement of width and length. In the statement, the leaves have different measurement of width and length. A variance would be applied to determine how much variation is observed in terms of width and length. The square root of variance is standard deviation, which are expressed in equal units. It is measure of how far the average value of the set is from the average. The smaller the standard deviation, the closer the data points are to each other. In comparison with the spread of 2 different sets of data, which each have its own unit and measurement, standard deviation is useful. The last question is why it is necessary to have different sets of measurement for the measurement of leaf width and length. Having different set of data of measurement for the length and width of the leaf is useful in data because it is allow for comparison between different set of leaves and to assure a consistent result. It is necessary to make assumption about distribution of the leaf and relationship between width and length. By having different sets, we can compare our result with the other sets. This is done to ensure that the experiment produces an accurate and consistent data.
In conclusion, this experiment was able to demonstrate the basic concept of statistic and variation. We got the correlation R is 0.68, which is a strong positive correlation, and the p-value is smaller than 0.005. so the p-value is significant.
The effect of that factor on the distribution and abundance of organism. In the present case, we are exposing a couple of quadrat, 1 meter times 1 meter. The student must also take into account the anaerobic factor of a study site and classify plant species in that area. The objective of this experiment is to introduce the simple concept of quadrant, second, to enhance the power of observation, third, to record the difference in plant distribution within the quadrant, and last, to infer from natural observation the possible factor that affect distribution and abundance of organisms.
This is the result obtained for this experiment. We have two quadrats here. For quadrant 1, there are 5 species that we found in this quadrant. The 5 species are Euphorbia hirta, Mimosa pudica, Digitaria didactyla, Desmodium guni, and Taurinia crustacea. The total number of individuals we found in this quadrant is 80. For Euphorbia hirta, we have 5 individuals. For Mimosa pudica, we have 1. For Digitaria didactyla, we have 18. For Desmodium guni, 52. And for Taurinia crustacea is 4. So the density of the plant is as shown as in the figure, where it is calculated by the formula. The number of individuals and is divided by the area of the plant. Second quadrant, there are only 3 species obtained in this quadrant. The 3 species are Digitaria didactyla, Desmodium heterophyllum, and Convolvulus farinosus. The total number of individuals found in this second quadrant is 18 individuals. 10 for Digitaria didactyla, 7 for Desmodium heterophyllum, and 1 for Convolvulus farinosus. The density of each plant is calculated using the same formula as the above in the quadrant 1, and get the density as shown in the figure here.
In this experiment, we use quadrat of 1 by 1 m square. We calculated the density through the formula of the total number of individuals per the measurement of the length. We find the term quadrat in practice. Quadrat is a square frame of 1 by 1 m square that is placed in a particular habitat of plants. The quadrat is useful for collecting samples of sampling plants. It is a designated area used to estimate population in an exact area by counting the plant in that area. What are the 2 basic requirements of all the quadrat techniques? The first one is the area of the quadrat must be known. It is essential to know the exact area of the quadrat being used in the sampling technique. The area of the quadrat is typically determined by measuring its dimension, such as length and width. Knowing the precise area allows for accurate calculation of population density or other ecological measurement within the quadrat. Second, the population in quadrat must be determined exactly. To calculate population density using a quadrat, the number of organisms or target sample within the quadrat must be determined precisely. This requires careful counting or identification of all samples belonging to target species of organisms within the quadrat. What would be the advantages and disadvantages of using a larger or smaller sample quadrat to survey the sedentary organism? The advantage of using larger quadrat is that it can give more accurate estimation of overall densities and allow us to sample species population spread out over a large area. However, its disadvantage is the sampling process will become more complicated and need to spend more time to finish sampling. Meanwhile, the advantage of smaller quadrat is that it is very easy to use and does not take more time. But the disadvantage is can sample only a small range of species population.
For question number 4 about the issues concern ecologists in conducting sampling method due to spatial scale, this is the answer. We know that it's hard for us to estimate the densities of the large and moving organisms. So this is the issue that being discussed by the ecologists. All we want to do is to choose the correct sample area and also minimize the number of sample by having the great result with the limited resources and also time. For number 5, they're questioning why there are difference in number of species in a quadrat. For our assumption, the number of plant species may vary due to the adaptation of that plant. Means that higher adaptiveness plant may increase the population there compared to the plant that have lower adaptiveness to that particular quadrat. Besides, physicals and also abiotic factors also involved in these issues. For physical safety, for example, plants that have thorns that live in a dangerous area tend to survive more than plants that have no thorns. By this, plants that have thorns tend to have more population or more species compared to the plant that have no thorn. For abiotic factors, some plants may be affected by the presence of the wind because they cannot stand with that situation. And some plants may also cannot stand with the low temperature, which is the factors that affecting the distribution is the temperature itself. So the plants that have low durability to that particular temperature, which is the low temperature, tend to not survive in that quadrat.
Conclusion, the objective of this experiment was to enhance the power of observation and to record the difference of plants distribution in a quadrat. So based on the result that we obtained and the discussion that I have made previously for this experiment, we can conclude that this experiment is successfully achieved the objective, which is we found the species in the quadrat and we count how many species in the quadrat.